And now I'll turn the podium over to the moderator for our second session, Karen Lillycrop, another member of our planning committee. Karen.
Hello, everybody. So we're now basically going to turn that page and go on to chapter 2, which is about etiology and causal inference. And we've got 4 speakers. And again, what we're going to do is get them to speak first of all, and then we'll call them up at the end, and then we can have the questions at the end. Okay, so our first speaker in this session is Jacob Friedman. He is professor at the Department of Pediatrics at the University of Colorado. His primary focus is on the understanding of early life nutrition and the environment on molecular, endocrine, and epigenetic origins of childhood obesity. And today he's going to talk about epigenetic mechanisms for obesity risk. Jacob, thank you.
Thank you, Karen, for the introduction, and thanks to the organizers for allowing me to come to talk to you. This was my title, my given title, and I will have to say that the stage was well set by the previous 2 speakers, and so what I hope to do is talk to you more about exposures and outcomes that might be of clinical relevance to humans. So I have 2 objectives, pretty big ones actually. I'll try and cover them as quickly as I can, which is to talk about prenatal exposures, that is maternal obesity, how we can separate diet from obesity, talk a little bit about inflammation, talk a lot about how you get fat in the beginning and where the epigenetic risks may lie for subsequent childhood obesity. And I'm actually going to spend a lot of time focusing on the liver because that's where a lot of our studies are. In the second half, I'm going to sweep through the microbiome, and I know my colleagues here from Seattle are going to do a much better job of this. It's a huge topic, but a very important one to talk about the metagenome. I'm going to talk about breast milk and risks for how the microbiome informs our disease pathways and maybe have some time for future directions. So these are the latest data from JAMA that show what are we up against. This is maternal obesity in the reproductive years. And if you look down here at all people surveyed across time, you see roughly overweight and obese is accounting for about 60% of the populations that we see that come into clinic. 33% are obese, and the super obese, the highly obese, are about 8.5%. If you look at health disparities, if you look at Hispanics and blacks, you get almost 80%. So this is what we're up against, the starting point. So I like to say, how could this not impact infant outcomes? Probably you could figure this out. So we know that obesity begets obesity. There's clear associations between maternal obesity and early onset obesity of children. There's evidence of metabolic syndrome. There's evidence of fatty liver, cardiovascular disease, and this is not news to anybody here. And animal and human studies have shown that there is aberrant methylation, and you can see this in animal models, and there's emerging data for methylation in humans. If you do liver biopsies, you can see this. This idea of mitochondrial bioenergetics, or how do we get obese? So where this might start might be down within our energy metabolism pathways, and I'll have a bit to say about that as we go forward. So I wrote this editorial back in 2011, Childhood Obesity, Methylate Now, Pay Later. And this was based on a study that came out that showed in cord blood, if they measured methylation status of specific genes in the human cord and subsequent development of childhood obesity, they could actually predict, or using some of these marks that would have maybe future utility in identifying future risks, but not necessarily were causative. And the issue is, what is causing these marks? Is it oxidative stress? Is it nutrient excess? Is it inflammation? We have really not too much of a clue. If you go back into the literature and look across time, this was 2014, it probably needs to be updated now. There's various maternal diseases, diabetes, obesity, and low birth weight famine that have been marked with these methylation marks across the genome, and a lot of times this is done in placenta and blood. So we talked a little bit about tissue specificity. There's probably some of this going on, but we're not exactly sure how much. It is probably possible to identify differential methylation of thousands of genes across these metabolic pathways, but really it's about whether it's cause or effect, and whether it's genetic variation as well as epigenetic variation. I think we need to think about that. So the way I think about this is, in a big way, is how do we solve these problems using the modern tools of omics. And so we're really interested in what you're eating and how mother's metabolome informs her microbiome and epigenome, because we believe it's environment, obviously, that's affecting the epigenome, and not just mom, it's the placenta. So the transcript in the placenta, shown here, can be highly different across lean and obese patients. This is an example of some inflammatory pathways from obese mothers, showing that a lot of genes are turned on early in the placenta. And the neonate, of course, derives its signals from the placenta, and its epigenome, both imprinted and non-imprinted, comes from whatever gets across that placenta. And the outcomes then play themselves out in terms of establishment of its own metabolome and epigenome, and ultimately proteome. So our work clinically has touched on all these areas, and I'm just going to highlight a few of them here, in which what we're really interested in is how these obese women come into pregnancy, what they're eating, can we fix their diets, does it affect the fuels that cross the placenta, does it affect adipose tissue mass, inflammation, and production of fuels, and more importantly, what comes across the pipeline here in the umbilical cord. So we can actually, and I'll show an example of this, of the mesenchymal stem cell derived from newborn infants exposed to maternal obesity. And then using MRI, we can actually take a closer look as to where is the fat, how is it accumulating, and what happens to it. This is just a happy example of the lab. So actually, Kristen is here in the audience, and Allie is also here, and they're harvesting some stem cells. We're putting infants in a pea pod, and here's a shot of the MRI to get a look at the liver. So one of these things about where does the methylation occur, is it across tissues, and is it permanent? So this is a really novel technique that we've just started in the lab, in which we take the umbilical cord, and within the umbilical cord, there's a population of stem cells called the mesenchymal stem cell, and they live and they get exposed to all the things that the fetus does. And it's exposed to transplacental blood, and these stem cells can be harvested and differentiated, and you can study them in vitro, either as myocytes or as adipocytes under exposure of a variety of conditions. And so this is just the example of 3 pathways that it could form. So maybe the idea is that these early exposures could program infants more towards the adipocyte lineage as opposed to the myocyte lineage. It's non-invasive, it's plentiful, and its population is available to epigenetics. So here's a brief shot of what we've been doing, looking at normal weight and obese weight or obese women. The maternal characteristics. This is Kristen's work shown up here, in which the pre-pregnancy BMI is quite high in the obese women. They have higher HOMA-IRs, they have higher lipid levels, and all these things are crossing the placenta. So the hypothesis is that infants born to obese mothers may be predisposed to early onset metabolic disease due to programming events before birth, because of fuel overload or intramitochondrial pathways associated with energy metabolism. So it would change fatty acid oxidation, cell differentiation, and intermediary metabolism, and ultimately epigenetic mechanisms are testable. This is just one example of what we see, and this is a paper that's, or an abstract that's going to be presented in the ADA, in which you look at demethylases within these adipogenic and myogenic derived cells, and they're both sort of suppressed here, DMT1 and KMDA6A, and we are actively studying the role of these and their ability to regulate metabolism in the cells. And if you look at the GLUT4 content, which is a marker of insulin sensitivity in some ways, babies born to obese mothers, shown over here, have lower expression of this GLUT4 mRNA in this stem cell. So I'm going to switch gears and tell you about our second story, and this relates to collaboration that Kevin and I started many years ago, and I'm not going to steal his thunder, but to talk about is it diet? And if it's diet, is it a matter of fat? So with at the Oregon National Primary Research Center, with a host of people, we've worked on developing this overfed model in order to examine the effects of maternal obesity and later on diabetes on the development of these metabolic systems. And so this is one of the key findings that came from these studies. And what's going on here is we've got two pods of animals, some fed a chow control diet and others prior to conception given a high-fat diet. They're mated, and then at the early third trimester, you can perform a C-section, and this is what we see in the liver. The liver is chock full of lipids, and this is not benign. The livers are actually making oxidative stress here as indicative of 4-HNO1E. And so just the exposure to the diet, we asked the question, if you change the diet, so what we did was we reversed it, put the animals after C-section on a healthy diet, and the next spring took the animals out, and what we saw was a nice reduction, but not total reversal of the fetal liver triglycerides. And so the liver, we think, is one source of disease pathology, but Kevin will tell you that this is hitting every single organ in the animal, its brain, its pancreas, it's a lot of things. Does this happen in humans? So what we did here was we took babies born to obese gestational diabetic moms at 2 weeks of life and put them in an MRI. And what's shown here is an infant, it's breastfed, it's wrapped up in this special papoose. It's listening to its favorite iTunes over here. And we used an intralipid, a bottle of intralipid as an internal standard. And this is Dave Brumbaugh's work where he took 13 infants born to normal weight and 12 born to obese mothers. And in this MRI, what you can do is look within the liver, the ratio of water to fat, and estimate how much fat is in its liver. And this was published last year in Pediatrics, and it was really, to me, it was sort of the surprising thing about where things are in terms of fetal life. Hepatic lipids in these babies born to obese GDM mothers is higher, about 72%. But what was interesting to me was that it was the maternal pre-pregnancy BMI that actually predicted, and not the infant fat mass that predicted how much liver fat there was. And there was very, very little visceral fat, unlike you and I sitting out there where we have, if we're developing fatty liver, we have lots of central obesity. The baby is not born with central obesity. What this means to me is that the fuels on board from mom who is obese have nowhere to go but into the liver, and it's really her obesity and not the infant's obesity that's probably really important for whether this child is born with a fatty liver. So we now recognize that this is about, in our clinics or in childhood GI clinics, there's a high risk of this fatty liver disease in kids who are obese. Probably about 55% of obese kids have fatty liver the first hit. Some of this could be due to genetics, maybe epigenetics, some of it due to risk, and some of it obviously due to lifestyle, things that we're doing to ourselves in our environment. This second hit is where I want to talk to you about why does this progress and how does this relate to diet and other exposures that might be involved in the microbiome. So in order to get to the more severe form of NASH, you have to have oxidative stress and hepatic injury. And where this occurs is through inflammation, which we don't think occurs in utero, but it happens to occur as a result of the microbiome. The question is this reversible? So last year we published this study where we took these animals and we weaned them onto a healthy diet. So these are offspring of the control and the high-fat diet. And at 7 months on a healthy diet, we looked at them at a year out, and what we found was that the juvenile livers from the animals that were born to mothers on a high-fat diet but were following a healthy diet had these elevated de novo lipogenic genes activated, and a lot of these pathways are known to be methylated in fatty liver disease. And more importantly, when we look back at who the mothers were, we found that this was only happening in the high-fat, insulin-resistant mothers. So the mothers that gained the highest amount of weight and were highest in insulin resistance bequeathed the juvenile fatty liver to the offspring later in life. Molecularly, sensitively, at the level of where this happens, we know that these fuels cross the placenta. They probably overwhelm the mitochondria. The genes for de novo lipogenesis are not expressed in utero, but the mitochondria, the result is that there's a lot of oxidative stress and a lot of loss of control of sirtuins that control the activation of these genes. So these are acetylases or deacetylases, and we've talked about methylation. We haven't talked much about acetylation, but these seem to be downregulated in the cells, and it can lead to oxidative stress, inflammation. So now I'm going to tell you why we think the microbiome is important. So you all heard about the what's called dysbiosis, or people who are obese have a dysbiotic microbiome compared to people who are lean. What does this mean, and how does it occur, and when would it occur in an infant? So we know that the neonatal period, right after birth, is critical for programming. I've talked to you about what happens in utero. Now, during breastfeeding, you get exposed to these novel nutrients, lots of bioactive molecules, bacteria. You double your fat mass in the first 4 months of life. Okay, so here's the second period. The brain neurocircuitry is being established. You're establishing your immune system, a symbiotic relationship with the gut, and you're getting instructions from your diet in the early aspects of immune protection. So what things can you let in and things that you can't let in, and how does this maybe program your inflammatory pathways for maybe the second half of what's going to happen to you in life. And part of this has been shown to take place at the level of epigenetics. So things that are in the microbiome really can modify the immune cells in the gut, and other speakers will talk about that. So the way I think about this is as follows. If mom is on this Western-style diet that we feed the animals, for example, she is now breastfeeding an animal or a child, and this, not the animal, it's a child. So what happens here is there's a dysbiosis in mother, which I'll talk to you about in a little bit, and so the infant is feeding on the diet, and its gut is quite leaky in early development. So these microbial products that are setting up shop down in the infant gut frequently can come across the gut, and they have a direct highway, a pipeline right into the liver, okay? And so I've told you about development of steatosis. What I think is happening here, our idea is that there's epigenetic programming, and this probably takes place within the macrophage itself to drive this disease from just simple steatosis to NASH. And so if you back up the pipeline, it's really what's happening during feeding and breastfeeding. This is how a remarkable paper that I think bears a lot of discussion, and this is by Corin et al. This shows you that the microbiome in a mother who is normally pregnant, if it's transplanted into a gnotobiotic mouse with no bacteria, the mouse is normal. Same woman, in third trimester, you take her bacteria from her GI tract, transplant it into the sterile mouse, and you get a fatter mouse with insulin desensitization. So this has me thinking quite a bit about the next half of programming. You can imagine that nature has made a system so that you can retain energy as you become pregnant in order to grow a fetus. More importantly, you transfer those microbes to the next generation, and the baby needs to hold on to these microbes in order for it to grow. But I think our cause for concern is women who come into pregnancy with an already disordered microbiome and undergo this process, what happens to the infant? This is an example in the primate what happens. So my colleague Kirsty Agard took these animals, as I mentioned, they were fed or exposed to this maternal high-fat diet, then switched to a healthy diet. And what she found was that the maternal diet during pregnancy persistently altered the offspring microbiome, shown down here. Okay, so that even on a healthier diet, you're getting the microbiome from the diet that your mother gave you. So we have been tackling this in humans, and I'll just show you a couple pieces of data to end this talk. We think that the maternal obesity will directly affect the development of the infant microbiome and be associated with increased adiposity during the first 4 months of life. So the question is, do your genes act on your microbiome, and which in turn, therefore, will promote disease? And so it's really about what's known as the undiscovered country within down here. So we have this grant to look at this in 4 phenotypes: normal weight, overweight, type 2 diabetics, and gestational diabetic women entering gestation. And then the babies are all delivered vaginally. They all, none of them are antibiotics, and they all agreed to breastfeed at least for the first 4 months. And we're assaying the maternal microbiome and the infant microbiome, and we're using this to infer causation for adiposity. And so our first look is what are they feeding on? So breast milk, we know, is a potent driver of the microbiome, and what's in breast milk is presumably protective of obesity, but we really don't know a lot about what's in obese breast milk. It's driving appetite, inflammatory cytokines, it could be having antioxidants, different fatty acid profile, and affecting the infant's microbiome. So we've looked at this, and there's a whole host of things that you think about in maternal obesity are in this breast milk, including some microbes that are believed to have, even though they're very low abundant, might be patterning the infant. But what we've seen in mothers who are obese is over here. The amount of triglycerides in maternal milk is not different, so the macrofuels are not different according to maternal BMI. But what is strikingly different is the levels of leptin, insulin, and the pro-inflammatory fatty acids that the women are eating. And this is evident here, showing that if you assay what's in milk for the N6 to N3 fatty acid ratio versus what's in blood, this is a biomarker of diet, there's a very striking relationship between how much the red blood cell will tell you're eating and how much is actually turning into human milk. And some of this can be used to predict fat mass in the infants at 4 months of life. So one piece of microbial data that's emerging is shown here, and we've used breast milk, leptin, insulin, and the N6 to N3 fatty acid ratio to actually predict, either positively or negatively, the early infant enteric microbiome. And what you can see with these statistics is that some of these are being driven by insulin, and some of these are being driven by N6 to N3 fatty acids. So again, the concept is that the exposure from the diet through breast milk is patterning the early infant microbiome and might be playing a role in inflammation and weight gain. It's as such. So these bioactive components inform the gut taxa, which then inform metabolism, which then can make infants more prone to inflammation and epigenetic programming down the line. So there's a lot that we don't know in this field. We don't know about what's causing the colonization over time. This community, does it solve for a habitat-specific metagenome? And then this differentially regulates itself. And what is the time scale for this, and how does this occur? I should also mention that the molecular function of the genes in the bacteria are not the phylum. This is not the microbiome. The core human microbiome is this. It's the pathways that they make. And so over 2/3 of the genes are actually uncharacterized, more than any other single bacterial genome. We don't know the cell types and how they communicate. So I'll leave you with some final thoughts about this. It's that humans share this core microbiome, and yet they differ by gene, species, ecology, gene count, and richness. The microbiome is dynamic. We don't know a lot about the time scales. The changes in diet that can lead to short-term changes in the microbiome, yet it's not clear which of these are reversible. As I showed you in the primates, some of them are, but most of them aren't. The gene richness is a key stratifier for the response to dietary intervention, yet when you put these genes into mice, they become obese, but eventually they revert back, so it's not permanent. And some of these microbiome-derived metabolites have a positive effect on inflammation and energy harvest, while others are more toxic. So I think the key here is, can we identify the specific species or patterns of the microbiome that are more relevant as targets for obesity? So when I think about this, when I think about epigenetics, as we've heard about, We don't know what the causes are, but for low or neutral risk, we know that things like exercise, low inflammation, and normal gestational weight gain are probably important. For increasing your obesity risk, we know that things like smoking, gestational weight gain, high-fat diet, and insulin resistance are probably important. I highlight the fact that there are new genes. So if you read the latest issue of Nature, now we know there's a whole host of obesity genes and type 2 diabetic genes that probably are new targets for epigenetics. On this side, when you think about the microbiome, I think it's a potent area for discovery because there's a lot that's just not known. So things like altered short-chain fatty acids, low diversity, high leptin, all of these things through the first 1,000 days of life probably contribute together to drive obesity development. So I'll just stop there and say thank you.